being here for this TMS invited webinar, which is the last of the season. Since the next Tuesday, I remind you that we hold the most important event for the TMS, which is the Day of Science. Don't miss it, we have many important speakers, including a Nobel Prize winner. Today is my great pleasure to introduce you our speaker, Professor Anna Teti, the chair and the founder of the TMS. So just a few words about her career. Professor Teti is currently a full professor of histology here at the University of L'Aquila, and over the years she received many international awards, including the Chemofux Prize, the Prix and the Liquids, the Swiss Bridge Award, the SCTS and Mike Horton Awards, and the ASBMR Lawrence Race Awards. She is also the past president of the European Calcified Tissue Societies, the most important European calcific scientific society dealing with bone and calcified tissues. Professor Teti published over 200 papers. She has an H-index of 56 and almost 1,000 citations. Her research over the years mostly focused on bone cells, osteoporosis, mechanobiology, breast cancer, bone metastasis with focus on tumor dormancy, and autosomal dominant osteopetrosis, for which she is developing an experimental therapy based on CNS. Moreover, Professor Teti is a member of the editorial board of many international journals, and she is a reviewer of more than 40 international journals and 15 grant agencies. Today, Professor Teti is with us to talk about communication between cells in the bone, which I'm sure is a topic everyone will find very interesting. Finally, I remind you that at the end of the presentation, you can ask your question on by raising your hands or through the chat. So, Professor Teti, thanks again for being with us today, and the virtual floor is yours.
Thank you, Pier Giorgio. Too nice, this introduction. Many thanks. So today I'm going to talk about communications of cells within the bone, which is a very complex tissue, as I will show you later. So as you know, there are many means by which the cells communicate between them. And I would like to mention here the endocrine communication through factors that are released in the bloodstream, paracrine communication through factors released in the microenvironment, communicating junctions that are communication that requires communicating conduits, Juxtacrine communication that requires contact-dependent signaling, and extracellular vesicles mediated communication that requires release of membrane-bound vesicles. So why the bone needs such an important communication, complex communication? This because the bone is not a standing alone tissue, but is associated with the bone marrow. So this is a complex organ that put together two different, apparently different tissues, and in which there are many cells and tissues that coexist in the same environment. So it is absolutely important that the many cells within this environment communicate each other. Why this? Just to give you some examples, cells within the bone microenvironment must communicate for many reasons. This includes, for instance, the transition from cartilage to bone, because most of our skeleton starts as a cartilage, and then this is replaced by bone, as you see here in blue, the cartilage, and in red, the bone. This is important also for the skeletal development and growth, for the skeletal modeling, renewal, and repair, and for the bone marrow homeostasis. So in the bone microenvironment, we have many types of cell-cell communication. For instance, signaling across gap junctions, paracrine signaling, juxtacrine signaling, and signaling through the extracellular vesicles. So I'm going to touch all of them during this talk. I will focus only on the bone cells, not on the bone marrow, which is by itself a very complex system in which we have osteoblasts. This is the bone matrix. We have osteoblasts that form bone. Then they become bone lining quiescent cells or osteocytes that remain entrapped in the bone matrix. And on the other side, we have the osteoclast lineage that derives by fusions of specialized macrophages. So all these cells have to work in a concerted manner, and communication between them is absolutely essential for this reason. Let's start with the paracrine communication. This is a very complex figure, but just follow what I'm saying. In violet here, you see the bone matrix. These are the osteocytes with their processes. These are the osteoblasts, and this is one osteoclast. As you see, the osteoclast is a giant cell and is polynucleated. With regard to the paracrine communication, I want to focus on two of them that are really important for the communication between the bone cells. One is the RANK ligand RANK axis, with the RANK ligand that is produced by the osteocytes and RANK that is the receptor expressed by the osteoclast lineage. Also, the RANK ligand is produced by the osteoblasts. And together with the RANK ligand, these cells also produce the osteoprotegerine that is a RANK ligand decoy receptor. So this is one of the major pathways that stimulate osteoclastogenesis. On the other side, I want to mention the sclerostin DKK1 pathway. Just focusing on sclerostin, it is produced by the osteocytes, and this inhibits osteoblast activity. So it's an inhibitor of bone formation. So these two types of communication are very important for the balance between the bone cell activity. I want just to show you how these two pathways work. Regarding the RANK ligand, you see here the osteoblast and the lining cells, the bone just formed and mineralized, and these are the osteocytes on this side. So the osteoblasts derive from precursors that derive in turn from mesenchymal stem cells. And they produce both the RANK ligand and the co-receptor osteoprotegerine. While the osteoclast precursors, when they are still mononuclear cells, they express RANK. You see, the RANK ligand can bind the receptor RANK and they stimulate osteoclastogenesis. So it's a pathway that induces osteoclast formation that is counteracted by the osteoprotegerine. On the other side, the osteocytes produce the sclerostin, and the sclerostin works on the LRP5 receptor pathway, blocking this activity, thus reducing osteoblast function. These two major pathways have been very much investigated in the bone field, and what we know now is that there are important translational implications from these studies. For instance, they produce denosumab, that is a RANK ligand neutralizing antibody that blocks RANK ligand produced by the osteoblast and inhibit osteoclast formation. So this is useful when the osteoclasts work too much. On the other side, when these two pathways are blocked, for instance, in genetic disease, then osteoclastogenesis is impaired. And this is the case of the so-called osteoclast-poor osteopetrosis, in which, they say that it's poor, but indeed, osteoclasts do not form at all in individuals affected by this form of osteopetrosis. If we look at the sclerostin pathway, also here we have translational implications because Romosozumab is an antibody that blocks sclerostin. So in this way, it prevents the binding of sclerostin to the LRP5 Wnt pathway, and in this way, there is an increase of bone formation. So the studies translated now in therapies for patients, and also in this case, if there is a mutation, for instance, in sclerostin, then you have the so-called high bone mass syndrome, in which the bones are too heavy and the renewal is prevented. Okay, regarding the communicating junctions, these are junctions between cells that require cell-cell contact. In this imaging here, you see the osteoblast, the bone surface. This is an osteoblast that is becoming osteocytes. And here you see the osteocytes, which have a cell body and the number of processes that go into canaliculi and touch each other between the osteocytes and also between the osteocytes and the surface osteoblasts. This is important to keep the cells alive, even if they are very deep into the bone. Here, for instance, these osteocytes are very far from the blood that is here in this channel, and they are separated by this part of the system that is associated to the channel through a cement line. But still, you see that there is cell-cell contact between the osteocytes that are external to the cement line and the osteocytes that are internal to this cement line. So there is this tight communication, which keep also these osteocytes that are very far from the blood alive. What is the system by which these cells communicate? So you see here a process from a surface osteoblast. This is newly formed matrix that is still unmineralized. And here there is the process of an osteocyte that is deep in the mineralized matrix here in dark. When these two processes meet each other, they form gap junctions. So the gap junctions are made by connexins that are one against the other in the two adjacent cells. And these connexins have a channel. So this channel is open on both sides. So therefore, small molecules can cross the channel and go from one cell to the other by diffusion, so according to the gradient. So in this way, there is an exchange of small molecules such as ions or small second messengers, for instance. So it's a direct communication between the two cells that are associated through the gap junctions. What happens in the bone? In the bone, the subunit of the connexin is connexin 43. This is the major connexin expressed in bone cells, and you see here how this protein is made. A connexin is made by 6 subunits of connexin, in this case 43, that form a transmembrane channel that allow the communication of the cytosol with the extracellular matrix if there is only one channel. This is called the hemichannel. If two hemichannels are in contact between them in the same site, then you have a single channel that induces the communication between one cell with the other, as I showed you in the previous slide. So these gap junctions, they form a canal network between the osteoblasts and the osteocytes, between the osteocytes and the osteoclast, and here is not depicted also between the osteoblasts themselves. Whereas the hemichannels are typical of osteoblasts and osteocytes and induce the communication of the cytosol with the extracellular matrix. So there are stimulators of the gap junctions, such as the hormone PTH, parathyroid hormone, mechanical forces induced by fluid flow, prostaglandins, interleukins, and second messengers, such as the cyclic AMP. And there are also specific stimulators of the hemichannels, including, again, the mechanical forces by fluid flow, drugs such as bisphosphonates and calcium. So let's move now to the Juxtacrine communication, which requires a ligand receptor pathway that mediates the communication in a way that the cells must be close to each other. And for this, I'd like to go back to the RANK ligand pathway. That is also, the RANK ligand is also a transmembrane protein. It's not a soluble, only soluble, but it's also associated to membranes of the stromal cells and osteoblasts, and also T cells. And you see here where the RANK ligand is located in these cells. And on the osteoclast counterpart, you see the RANK receptor that binds the RANK ligand. So in this way, this pathway works also by a Juxtacrine communication. And what it does, again, is the activation of the osteoclast activity. And you see here, it is sensitive to some stimulators, including the parathyroid hormone and the vitamin D3 that are essential for the bone homeostasis. There are other type of Juxtacrine communication. For instance, the Ephrin pathways. In the bone cells, we know two groups of Ephrin pathways. The Ephrin B1, B2 with the receptor FB4, and the Ephrin A2 with the receptor FA2. Actually, both ligand and receptor transduce the signal. So it's two-way signaling. So we can consider both receptors. And what happens is that for the Ephrin B pathway, Ephrin is expressed by the osteoclasts, differentiated osteoclast, and the FB4 by osteoblasts. And when they bind, you will see an inhibition of osteoclast differentiation and the stimulation of osteoblast differentiation. On the other side, the Ephrin A2 and the FA2 receptor works the other way around. So when they bind, then you see a stimulation of osteoclast differentiation by the precursors and an inhibition of osteoclast activity. So all these Juxtacrine pathway harmonize the activity of osteoblasts and osteoclasts. And now let's go to the extracellular vesicles mediated communication. Extracellular vesicles are known since many, many years, but just a few years ago, they became very popular as a means of communication, I would say, among virtually all cell types. And so they are very popular, and there are a lot of work on this topic in the literature. So what are the extracellular vesicles? They are membrane-bound vesicles that are classified in various types. I mention here the microvesicles that originate by budding and then shedding. They have a diameter between 100 nanometer and 1 micron and have a cargo that is made by surface proteins, cytosolic proteins, and nucleic acid. And so in this way, they are released from the source cells. There are also smaller vesicles called exosomes. The diameter is between 50 and 100 nanometer. These are made by the early endosomes that internalize a number of molecules, including specific exosomal proteins, cytosolic proteins, and small RNA, including the miRNA. So this internalization form what is called a multivesicular body. Then it's released by exocytosis, releasing also the single exosomes. And finally, we have the apoptotic bodies that are made by cells by blabbing when they undergo apoptosis. They are large, 1 micron to 10 micron, and they contain especially genomic DNA. So the way how these vesicles communicate with the target cells is depicted here. So this is the secreting cells with the various type of vesicles, and they act on the target cells by fusion, by endocytosis or phagocytosis and processing, and also simply by surface interaction. So there are many ways by which these vesicles work on the target cells. In bone, it is known that the vesicles are absolutely very important for the crosstalk between cells, including osteoclasts and osteoblasts. And you see here a number of vesicles that regulate both cell types, and this here some of the cargo molecules that have been recently identified in these molecules. But these cells also communicate through vesicles that are released in the bloodstream. And in fact, as a consequence of the EV studies, very popular is now the so-called liquid biopsy, in which the circulating extracellular vesicles can be collected and analyzed in terms of their content in order to identify, for instance, diagnostics or prognostic factors. We have investigated the EVs in the bone, and I will give you some examples of what we have done. Especially working on osteoblasts EVs. The first observation that we made was that when we culture osteoblasts that are labeled in their membrane, their surface, with this red dye, and if we culture them in transwells on the membrane that separates these cells from unlabeled osteoblasts that are plated in the bottom chamber, lower chamber, then what we observe is that at the very beginning, only the upper cells are labeled in red, whereas the bottom cells are dark. And here see by phase contrast that the cells are here. But after a while, here after 48 hours, you see that the cells in the lower chamber are now labeled. So this suggested us, since we have labeled the plasma membrane of the upper osteoblasts, is that vesicles have been released by these cells and targeted the lower osteoblasts. So we collected these EVs from the conditioned media of the osteoblasts, labeled them this time with a cytosolic, in this case green, fluorophore. And you see here that we really collect vesicular entities as shown here in this slide. And then at the transmission electron microscope, we observed the typical size and shape of extracellular vesicles. And they also express a typical extracellular vesicle marker, that is the CD63, poorly expressed in the whole cells, but enriched in EVs, both freshly isolated EVs and also EVs that have been frozen and thawed. And this is important when you have to collect a high number of these EVs, for instance, for treatment, that you cannot collect all together, so you have to freeze them in some way. So then we observed that these EVs that we isolated from the osteoblasts and labeled either with the red or the green fluorophore, red for membrane and green for the cytosol, when we cultured other cell types in the presence of these labeled osteoblast EVs, we could observe that these cells became fluorescent. Here are human endothelial cells, and here are primary mouse monocytes. Not only that, we also observed that the target cells incubated with our osteoblast EVs, labeled EVs, can integrate these EVs in their cytoplasm. And after a while, you see that distribution of green and red in the cells is completely changed, suggesting that they are say, sent to different compartments in the cell. So there is a kind of processing of these EVs. Since the RANKL ligand pathway is absolutely important for the activity of osteoclast, we also investigated if this RANKL ligand molecule is present in the osteoblast EVs, and indeed this is the case. About 50% of EVs are RANKL ligand positive. This number increases with PTH, that is a classical inducer of RANKL ligand in the osteoblasts. These EVs can affect, from osteoblasts, can affect the osteoclasts. These are osteoclast cultures controlled or treated with EVs. This is a staining specific for the osteoclast, and I remind you that the osteoclasts are giant cells. And you see here that there is a higher number of nuclei per each osteoclast, so they are bigger, and in fact, the area of this osteoclast is increased. So there is a kind of effect of osteoblast EVs on osteoclasts, and don't forget that the osteoclasts are very sensitive to the RANK ligand. Also we observed experimentally that RANK ligand is absolutely important for, in this case, osteoclast survival, because we treated the osteoclast culture with EVs from wild-type osteoblast or osteoblast that we collected from RANK ligand knockout mice. And here we had the trap staining, so the stain specific for the osteoclast. These are the osteoclasts, and in blue you see the nuclei. And in these cultures, we measure the number of healthy cells with many nuclei, the number of cells of osteoclasts that lost the nuclei, so they were suffering, and also the number of retracted osteoclasts also suffering. And you see here that when the cultures are treated with wild-type EVs, from osteoblast, wild-type osteoblasts, you see that most of osteoclasts are in good shape, whereas this number decreases in cultures treated with RANK ligand knockout EVs, whereas the number of suffering cells increased. These EVs also integrate in ex vivo and in vivo. This is a culture of the whole parietal bone, the calvaria. In blue, the nuclei of the cells, and in red, EVs in which these calvaria were incubated. And here, this is an in vivo osteoclast in which we injected Osteoblast EV labeled in red, and then investigated at these time points the presence of the label in the different organs. And you see here that the bone in red is the one that peaks very quickly, and then it goes down in a plateau that lasts several hours, whereas the peak in all other organs is negligible. Only in liver we find a later peak, so probably this is a kind of clearance of the material coming from the other organs. And given this distribution in vivo, we also investigated if we could improve the osteoclast phenotype in RANK ligand knockout mice. So these mice lack a RANK ligand, and they have no osteoclast at all. In fact, using this specific staining for osteoclasts, you see here that the untreated bones are completely unstained. And here you see that the stain appears when we injected these RANK ligand mice with wild-type EVs from osteoblast, and increase in with the number of EVs injected, with an increase of the number of cells that are trap positive. We found a decrease in a high number treated osteoclasts, but there is a reason, because the cells fuse, and you see here larger osteoclasts in with this treatment. Others have shown that not only the osteoblasts release RANK ligand positive EVs, but osteoclasts release RANK enriched EVs. So this release the RANK through the EVs can block the normal interaction between osteoclast and osteoblast through the RANK ligand pathway. So it works like a decoy receptor, and then it is internalized by the osteoblast and processed. So it's a way to counterbalance the activity of RANK ligand, similar to the OPG that I showed you earlier. Now, can we use these EVs as carriers for antiresorptive drug? The answer is yes. This is in vitro, in which we treated osteoclasts with two drugs that are very much used in bone, the zoledronate that induce osteoclast apoptosis. This is the free drug, and you see here that the osteoclasts are suffering. And this is the dasatinib, that is a tyrosine kinase inhibitor, and also you see here that the free drug disrupts the cells. And here you see the same drugs encapsulated in EVs, zoledronate and dasatinib. And you see here that the number of cells that are suffering with the maximal effect with the free drugs at 10 micromolar are suffering also when the drug is encapsulated in the EVs. And this is true also in vivo. We have a quick system to increase the osteoclast number in the bones. Just focus on these panels. This is the staining for the osteoclast that I showed you before, and you see here that the treatment with retinoic acid increased the staining compared to the sham, and treated with PBS and DMSO, we found no difference. These are the vehicles. And you see here that zoledronate reduced this staining. Dasatinib also reduced the staining. This is the EV alone, nothing happened, and EVs with zoledronate and dasatinib. So again, a reduction in osteoclast number. And this is just to show that only the zoledronate, either free drug or encapsulated in EV, induces apoptosis in the osteoclast, but this is absolutely known. This is just the quantification. These are the controls. Zoledronate and dasatinib reduce the osteoclast surface, the osteoclast number, and only the zoledronate induce, apoptosis, whereas the other parameters are all affected not only by the free drugs, but also by the encapsulated drugs. Last but not least, in another work, we also demonstrated that the EVs are important also in bone metastasis because they are produced by osteotropic cells. They have a high content of different molecules and affect the bone environment in different ways. For instance, they affect the osteoblasts that in turn reduce their own EVs, and this affect, together with EVs coming from the tumor cells, pre-osteoclasts to become osteoclast and so destroy the bone where there is the cancer. And also they affect both the vessels, inducing angiogenesis. So all these increase what we call the vicious cycle by which the bone metastases disrupt the bone tissue. So in conclusion, what I showed you is that cells within the bone communicate through all communication systems known so far. Paracrine regulation is led by RANKL to regulate osteoclastogenesis and Sclerostin DKK1 to regulate osteoblastogenesis. Signals across gap-junctions induce communications of osteoblasts, osteocytes, and osteoclasts between themselves and with the surrounding microenvironment. Juxtacrine signals may reciprocally regulate osteoblasts and osteoclasts. EVs are novel ways to which bone cells communicate. They play multiple roles in bone cancer development, and some signals, such as RANKL, use various ways of communication to regulate osteoclast function, including paracrine, juxtacrine, and EV-mediated pathways. The original work that I presented has been done by this group. These are our funding agency, and we are very grateful to them for supporting our work, and I would like to thank you very much for your attention.
Okay, Professor Teti, thank you again for this very interesting topic and for this very interesting presentation. And now we can start with the discussion and with the questions. Okay, I don't see the question. I have a question, Professor Teti, just a curiosity. For example, how vesicles can be used to carry drugs in pathology, for example, bone disease, or in other case, for example, cancer disease, is possible?
Yes, there are a number of studies in this regard, but there are also many limitations because this is a material that we have to take from humans. And as ever, when you use human material to treat other people, there are always problems to take into account to prevent any type of transfection, for instance, induction of infection diseases, or other type of reactions against antigens and so on. I'm confident that things will improve with time. I'm not sure that it will be an easy way to treat patients, also because you have to collect a very high number of vesicles. So I find this very difficult. What I can envisage for the future is maybe to make some kind of artificial vesicles that are produced with the right material to prevent this type of problems.
Okay, thank you, Professor Teti. Okay, we are waiting for the question.
Maybe it's easier if they raise the hand and ask the question. There's some messages.
Okay. Okay, in the chat we have the question from Antonio Maurizi, and he asks, why cells need to use vesicles to exchange materials instead of using other systems?
Yeah, Antonio, I'm not God, so I don't know what to say. But certainly is a way to send several types of messages to the cells. The vesicles are known, especially in cancer, since many, many years, but nobody at the time understood that it was also a physiological way for cells to communicate. I have no idea why this happens, but you can consider that there is a cargo, complex cargo with many types of molecules inside. So probably with these vesicles, you can activate or inhibit several pathways at the same time in the cells. Then I can add that, for instance, something similar that are called matrix vesicles are released by the osteoblasts, and they are not done for communicating with other cell types, but to induce the mineralization of the bone tissue. That, as you know, is a very peculiar mechanism induced only in the bone matrix or in other tissue, but only in pathological conditions. So probably we will know more about, so not only the communication between cells through the vesicles, but also communication between the cells and the matrix around the cells.
Yeah, thank you. This is Antonio. Thank you for the answer. Yeah, because so my question was related. What I mean, so I expect that if cells need to send a protein or whatever far from the primary site, I expect that they use vesicle in some way to protect the cargo. My question, so my doubt was only related to the local communication, so cells that are close to each other in the bone marrow or whatever. But so your answer makes sense in my mind. So
yeah, but it is also locally. You have to consider that the extra matrix is full of enzymes of various types. So it could be a way to protect this material, at least the material that is inside the vesicles, different for the surface molecules. So don't forget that the external matrix is not only structural proteins, but a number of regulatory proteins and enzymes, ectoenzymes.
yeah. Okay, thank you.
Thank you.
Okay, now we have Marco.
Yeah, I was just going to add something to this, just an idea, but in general to answer also Antonio's question and to discuss more. Maybe the point of a vesicle is that they have more than one molecule inside of them. So it's like if I communicate with you and I say a word, which would be in the analogy a protein, for example, if I send you a vesicle with 5 words in it, I'm giving you a more complex message. And so maybe this is the point of communicating with vesicles, communicating more complex messages all at once instead of sending 5 words separately, which could make sense, but you could receive them in the opposite way, and then the message is different. So this could be one of the reasons as well.
Well, I agree, absolutely. The problem could be that with a single drug you affect a specific mechanism, and you know what's going on with the vesicles. Maybe it's more complex. So I'm sure that we need much more work to use them, especially in the clinic.
Yeah, I agree also with the point of Mark. I would add also for the targeting, and not only for the number of messages. Could be because if you just release a protein in the microenvironment, it can be uptake by any cells, let's say. Why if you use a vesicle, you can target?
In fact, one of the reasons why we use the RANKL and positive vesicles is exactly for this reason, so to target the cells that you want to target. But again, as you know, I mentioned the osteoclast expressing RANK, but RANK is not expressed only by osteoclasts. So again, the specificity is a problem.
Okay, thank you. Then we have a question from Jacopo Di Gregorio. I see the hand.
Okay, hello.
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 Hello. That's just a curiosity that Marco's question made me think. Is there a competition for molecules that are carried by the extracellular vesicles, or is there a preferred protein that may be carried over?
Well, I don't think that we can say that there is anything preferred. It depends on the vesicles, the cell type, and maybe also the phase in which you collect the vesicles from specific cell types. So I would imagine in vivo, when you have the Autostrada del Sole in August, so full of cars that run in different direction, too many people. So it's a very crowded environment. So difficult to say. Absolutely, okay. In my opinion, I don't know. Since it's an open discussion, maybe if there are other people that wants to say something, yeah, absolutely welcome.
Because that may have some therapeutical applications, because if you can drive that competition, if that's present, you can force them to express the molecule that you want.
Absolutely, Jacopo, you are absolutely right. In fact, we are working in this direction. My concern is that, in my opinion, it's just an opinion, but all cells express almost all. It's just a matter of maybe of level of expression. But if you consider cancer cells, is there anything in cancer cells that is different from normal cells? Maybe something is mutated, but yeah, and so this is why it is difficult to have very, very, very specific targets. In fact, the side effects are always present with any type of drug. So the idea to encapsulate in EVs and send it to specific cells, that could help to reduce the side effects. I absolutely agree on that. Yeah, and you may ask, but they can use liposomes. But liposomes is not the same as vesicles. Vesicles are more complex. In fact, the liposomes generally they are recruited by the liver. So good for treating the liver diseases, but not good if you want to cure lung or kidney or others.
I got it. Thank you very much for your answer and for the talk.
Thank you.
Okay, we have again Marco.
Yeah, just to add one thing to Jacopo's comment. There are also some proteins that are known to be enriched in extracellular vesicles, such as CD63, CD81, the tetraspanins. This is not an exact science, though. So that if you attach, for example, a GFP to CD63, you will find GFP, you will find fluorescent vesicles, let's say, but not only fluorescent vesicles. So this is the point also that Professor was making.
Exactly. So, exactly. So in fact, the correct way to describe this is that the EVs for the CD63 or other thing is enriched. In the past, the reviewers were very precise. If you said that you had a purified something cell type or etc., they wanted to change in enriched. This means that they are always contaminated by other things. And I think that enrichment expresses better the situation, because you find more, for instance, CD63, as I showed you in the vesicles, but it's not that you don't find at all the CD63 in the cell.
Okay, now we have a technical question from Luca Giacchi, who asks what kind of techniques can we use to load drugs in extracellular vesicles?
Well, some drugs are permeant, so they get into, so just incubating them. For other drugs, people use electroporation, but this, of course, destroy a number of vesicles, so the yield is not that good. For if you want to load, for instance, with a dye or something, they can use esterified molecules that, in this way, they are permeant. They go into the vesicles, and there, if there are enzymes that can cut the ester residues, then it becomes non-permeant anymore, and so they stay entrapped. So depending on what you need and the type of molecules you want to put into your vesicles, with a balance between keeping them healthy and increasing the yield of the loading.
Okay, thank you, Professor Teti. Okay, Luca. Okay, see, write the
Thank
chat.
you.
Thank you. Yes, and yes, we have not other question in the chat, and I don't see the end. Okay. So I think that that's all. And Professor Teti, thanks again for her time and for her availability. And I remember that see you on Tuesday for the event of the Day of Science. Don't miss it. And yes, that's all for now. Okay.
Thank you.
Thank you, Professor Teti, again. See you on
Bye-bye.
Tuesday. Bye-bye.
Bye-bye.